have this MAL gene that codes for the MVP gene, maltose binding protein. I told you about this a few days ago. So the MAL gene is here on the plasmid. There's a promoter here called pTAC that's a really good promoter in E. coli. I'll tell you about that in a bit. There's also something here between the promoter and MAL-E called an operator, and I'm going to tell you about that in a minute. So we have pTAC, the promoter. We have an operator right here. You might make a note of that. Draw a little arrow between the pTAC and the MAL-E and put operator. Then we have our MAL-E gene that codes for the maltose binding protein, which is an E. coli protein. And then we've got our multiple cloning site region where we cut twice and cut out a 56 base pair piece of the plasmid, basically cut out the multiple cloning site region and replaced it with our GAPDH cDNA right there. And we cloned our GAPDH cDNA unidirectionally. And so we made this gene fusion between MAL-E and our GAPDH right there. And we did that, remember, by cutting with XMN1 right here to make a blunt end in our plasmid. And we cut with SBF1 down here to make a sticky end. We cut out this 56 base pairs in between and replaced it with our GAPDH cDNA. Now we believe we successfully replaced it with our GAPDH cDNA because on your gel you saw a PCR product of this region that was the right size had you inserted the GAPDH cDNA successfully in there. Remember, this was your 8-colony PCR, and you had plenty of colonies that looked like they had the exact right size in the plasmid. So we believe we're okay. We believe we're in good shape. And because we believe that, we've forged ahead and we're doing the protein isolation today. And we're sequencing this. We're sequencing it forward and we're sequencing it reverse. Normally we would sequence first and then do the protein isolation after we were certain that we had the right sequence. So we don't have time for that. So we're doing them both at the same time. We're going to sequence and do our protein isolation at the same time. But normally you would do that sequentially. Does that make sense to everybody? Okay. Now, we've made our fusion protein, we think. And now I'm going to show you how we're going to purify this fusion protein using a really clever system from New England Biolab. So this is shown on the next page, page 73. Schematic illustration of fusion protein purification using the pMAL system. Okay, this part of the diagram up here, this bar represents the plasmid. So imagine it goes from the left, it goes to the right, it extends down below the screen here, comes back around. So this is part of our big plasmid circle. This is the MAL-E gene here. Before the MAL-E gene would be that pTAC promoter, right? Then the MAL-E gene, which is a normal gene from E. coli that codes for that maltose binding protein. Then we've got our multiple cloning site region here that we've cut out and replaced with our target gene, which is the GAPDH, okay? And then after that, we're going to have stop codon at the end, which we'll look at later when we do sequencing that.
Is the target gene in frame?
Well, the way we designed the experiment, I'll show you this in a minute. The way we designed this experiment is clone the target gene in frame, and that's critically what we're going to look at with sequencing. Is not only did we get GAPDH in here, but is it in frame so that we make our fusion protein? That is, as we read through the triplets that code for MAL-E, we're going to get to the last triplet codon here of MAL-E, and the next 3 nucleotides better be our ATG of the GAPDH, which is our start codon for GAPDH, because that codes for the beginning of our GAPDH protein. So it has to be in frame. I'm going to show you that in detail in a minute. Okay. So we've cloned our gap pH in here, we believe, and now we've put them in E. coli, and over the Sunday, these were growing in E. coli. Our fusion protein was being made. I'll take you through the details of that in a minute. And here's our fusion protein. Our fusion protein is maltose binding protein, and then our target protein. And this little blue here is part of our protein that's encoded in the vector. So this is just schematic, but this little blue part is encoded in the vector. And that little blue part is actually a protease cleavage site. Yeah, I'll go back and show you this in more detail in a different diagram. So we have our maltose binding protein that ends with a protease cleavage site, and then our target protein, which in our case is gap pH. But this could be any other protein that you wanted to produce, right? Okay, so this is the protein produced from our gene fusion. Our gene fusion results in a protein fusion. Now, how are we going to purify this protein? Because of course E. coli is making this. It's making our protein, but it's also making thousands of other E. coli proteins, right? E. coli is growing and it's making all the proteins it needs for survival. So our protein is just one of many. So how are we going to purify our protein away from all the thousands of E. coli proteins that we don't want? Excuse me. Well, we're going to lyse our cells, we're going to get the protein out, and then we're going to load all the protein onto a column, and this column has on it amylose B, amylose protein. Amylose is a sugar that maltose binding protein can bind to. So maltose binding protein can bind maltose, but it can also bind amylose, a related sugar. So we pour our protein on the column, and maltose binding protein will bind to our amylose column with our target protein connected to it. Now you might say, okay, I told you maltose binding protein is a normal E. coli protein. What about the maltose binding protein that's on the normal gene on the chromosome of E. coli? And that gene has been mutated, so it's not making the maltose binding protein. Okay? So the only maltose binding protein we're going to have is from our gene fusion on the plasmid. Okay? So far, so good. We pour this on the column. The only thing that binds is our fusion protein. We wash, to get rid of all the other stuff. So we're going to wash a lot. And then we can lute out our fusion protein by adding into the column a high concentration of maltose. Maltose binds to the maltose binding protein better than amylose. So when the maltose binding protein sees the maltose, it lets go of the amylose and binds the maltose instead, because it likes the maltose better. And so now our fusion protein will come off the column, and we'll collect our fusion protein at the bottom. And we're going to run this fusion protein on a Western blot. We're going to run it on a regular protein gel so we can visualize it. We're also going to run it on a Western blot with a monoclonal antibody against gap pH to prove that we've got our protein. Now the last step would be to cleave at this cleavage site with the protease that cleaves that protease cleavage site, cleave away the MVP from our target protein, and then we would take this mix and put it on the column again. Not this column, a different column, but the same kind of column. Put it back on the column, and now the maltose binding protein will bind to the amylose, and our target protein will just flow through. Pretty cool, right? And so by running this column twice, we're going to get our protein purified out away from the thousands of other E. coli proteins. Super clever system, really clever. I've always asked around who developed this. I've never been able to figure it out, but if I ever figure it out, I'm going to send them a present. Because this system has worked great for us. And getting your purified protein at the end, even for non-protein people like us, works great. It's really foolproof, as you'll see. It works really well. Okay, so a very clever system. Any questions about the purification system?
The system works generically with any protein you might care about, or is there something special about our gas pH that allows it to work?
We've used this for proteins that we, clone the genes for, from parasites that we work on, from viruses that we work on, from mosquitoes we work on. So for us, this has been great. Now, I have heard from colleagues that this doesn't work for everything, and that's not surprising. And because this may not work for you and your protein, I'm going to show you some other systems. But this one has always been good for us. So whenever anybody asks me for a recommendation for an expression system, I always point to this one first. And then I say, if that one doesn't work, come back to me and I'll give you some other ideas.
Can you explain again how the molecule gets unbound from the MVP?
Oh, how the protein unbinds from here?
No, that I understand, because when you put the MVP and the protein again through a column, it needs to be
So when we get down here, we cleave with the protease, and this maltose binding protein, it bound maltose to get it off the column. It's not shown, but it's got maltose here. So what we have to do is dialyze to get rid of the maltose, and then once we've gotten rid of the maltose, then we put it back on the column, and now this will bind with the MVP will bind to the amylose, and then our target protein will come out. Yes.
So the code site, and then they add a little more codon at the end.
Right. Exactly. Yeah, I'm going to show that again. So, so let's back up to the previous diagram again.
Sorry, another question. Does this binding protein do anything to the secondary and tertiary structure of the protein that changes it, you get the kind of product out?
Does the fact that it's produced as a fusion protein change the folding of the target?
Yeah.
So I have not seen that, but I imagine that could be a problem, because you could imagine that bound to this other protein, it might not fold properly. Again, that's not been a problem for us, and it's probably that we're lucky that after we cleave with the protease, that it refolds into its functional configuration, but you wouldn't expect that to always happen. So again, I'm not a protein person, but people who deal with lots of proteins like this will give you protocols for refolding a protein, and then it might refold properly. Good luck. Okay. Let's back up and look at the fusion in a little more detail. Okay. This is our sequence for our multiple cloning site region, and before that is MALI, right? The MALI gene for the maltose binding protein. Okay, now we're going to look at that sequence down here in a little more detail. Here's the sequence of MALI coming in from this direction. Okay? So this is MALI out here. Then we have these codons. These codons are specifying the amino acids shown below. Right, so this is a single letter amino acid code. So we have codon, triplet codon, triplet codon, triplet codon, triplet codon, triplet codon, right, all along the end. Now, the XMN1 site, GAA, and then it's any four nucleotides, because remember it's GAA, N, and then TTC. So we have GAA, the first three nucleotides here, then any four nucleotides, in this case that's GGAT, and then TTC, right? And the cleavage is made right in the middle, so GAA, GG, and it cuts right here. So that's where XMN1 cuts. Right there. So we cut there. And then we also cut down here at the SPF1 site and release this. But what we really care about is this cut here. The XMN1 cuts right at the end of this codon. And so right there where my cursor is, that's where we want to ligate our start codon for GAPDA, right there. So because we cut with XMN1, we know we cut here, and if everything goes well, we should ligate at the beginning of our cDNA right at this point, right? Right at that point. What's our cDNA start with? Well, let's go, going the wrong way. Our cDNA, remember we PCR amplified with this primer that starts with the ATG, which is the start codon. So our PCR product, our GAPDH PCR product should start ATG with the start codon. So ATG is the start. So if we go back to that diagram, if we successfully cut right here with XMN1, then we ligate in our ATG right here, we should be in reading frame, right? Because we have a triplet codon here, and then we should have our ATG methionine, and then keep going. So if things went correct, the way we designed this by cutting with XMN1, which cuts right at the end of one codon, and then ligating in the beginning of our cDNA with the start codon, we should maintain that triplet reading frame so that we can get our correct encoding of all our amino acids. If we're off by even a single nucleotide here, then the reading frame is going to be completely off from that point, and we won't make our protein. We'll make something else, but it won't be our protein, right? Now, I'm going to take you all through this in greater detail when we actually get our sequences, because we're going to be looking really carefully at this position here to make sure that we ligated our molecules together. If everything went well, we should be good there. What possible things can go wrong? Well, there are nucleases around, and if our vector or our cDNA lost a nucleotide or two at the end, then our reading frame is going to be off. Have I ever seen that? Yes, I've seen that. But we don't see it often, unfortunately. So usually it goes together properly, but we'll see that when we get our sequence results. Now, when the protein is synthesized, when our fusion protein is synthesized, starting right here, it's not going to be MVP anymore. Now it's going to be GAPDH. So the amino acid sequence from this point on is going to be completely different because we will have ligated our GAPDH in here. And we know what that amino acid sequence should be because we know the sequence of our GAPDH. But it won't be these amino acids. It will be a different sequence of different amino acids, right? When we make the fusion protein, it should go right from this maltose binding sequence, with this being the last amino acid, going straight into our protein with its amino acids. And when we do the cut, it's with this factor 10 enzyme, which cleaves at the sequence IEGR. Okay, what's before the cleavage site matters. It doesn't matter what's after the cleavage site. So factor 10 will cleave here, no matter what we ligate in after this. There's some restriction. I think this can't be a proline, but I'll look that up. I forget what the restriction is, but there's one restriction. But having a methionine here is fine, and that's what we usually do. So it's no problem if you have a methionine codon here first. The factor 10 recognizes this sequence back here and says, aha, I'm supposed to cut there, and it cuts our two proteins apart. Okay?
The one delay we have still attached this amount of
Up to this point, going from way back here, there's a PTAC promoter back here, and then all the amino acids of MAL-E. The end of the MAL-E gene is right in here someplace. So it's functional MAL-E, it functions. The end of the MAL-E is here, and then it's going to start with our protein, and now it's going to be GAPDH amino acids all the way through after that.
But in addition to all the amino acids,
No. We cut all this out. When we cut with XMN1 here, we cut our vector here, and we cut it here with SBF1. When we did that, we cut out this whole multiple cloning site region. So that's not there. So we've essentially cut out this MCS region and replace it with a much bigger sequence, which is our GAP-PH. Remember when we cut with XMN1, it was blunt here, SBF sticky in here, cut out this fragment and replace it with our cDNA. Right?
With the small plasmid, are all those restriction sites designed to like have
No, they don't all do that, and so you gotta be careful what you use here. So every time I use this vector, I do it the same way, because the XMN1 cuts in a way that allows you to end with the MALI protein and then start with the start codon of your protein. So I like to do it with those two enzymes and take the rest of this out. You could cut with XMN1 and you could cut with pMAL instead, or EcoY1. At this end, it doesn't matter so much. But at this end, you want to cut with XMN1 because that's going to give you the right reading frame at the end of MALI and allow you then to clone in your gene of interest starting at the start codon. There are other ways you can do it, but for me, this is always preferred. And so that's the way I recommend doing it.
I think I'm a little confused about the terminology. Is XMN1 not the protein that does the cutting, or is that?
Oh no, these are names of restriction enzymes, sorry, so these are all restriction enzyme names. And so XMN1 is a restriction enzyme that cuts this sequence, and the E1 is a restriction enzyme that cuts this sequence, and so on. BAMH1, GGA, TCC. BAMH1 is a commonly used enzyme, EcoR1, GAA, TCC. These are all restriction enzyme sites, and those are the names of the enzymes. It's shown divided into codons so that we can see how this thing gets translated into protein. If you don't insert anything in here, then you get this sequence of amino acids, which doesn't code for anything, and then you just stop codon here. You see that's TAA, which is you are awful in the RNA, right? So if you don't put anything in here, you would stop at that point. That's where your protein would terminate. But we're putting in a whole coding sequence right here, and so we're going to make protein, and our protein sequence, our cDNA, ends with the stop codon. So ours will end with the stop codon from our gene, not that stop codon. That stop codon will be there, but that's not the one we'll use. Does that make sense? If there's anything confusing, when we actually get your sequences, we're going to look at exactly how this goes again. And sometimes it's easier to visualize everything if you have your own sequence in front of you. So we'll be doing that soon. As long as we've got this map up here, there's a couple other features I want to point out. This gene labeled LA is the ampicillin resistance gene. Ampicillin resistance will sometimes be written AMP with an R superscript, so AMP resistance, sometimes AP with the superscript R. The name of the gene is actually BLA, and it's BLA because it codes for an enzyme called beta lactamase, and so the name of the gene is actually BLA, which is funny, but on a lot of maps you will see that shown instead of BLA as AMP resistance. But that's what that is. The PTAC is our promoter for getting good transcription. Remember, there's this funny thing called the operator here, which I'll tell you about in a minute. This is another gene that codes for another E. coli protein. We don't use that in this system at all, so ignore it. This is our origin of replication here for working in E. coli. This is a plasmid origin of replication. Comes from a plasmid called PBR322. Not that you need to know that, but that's why it's called that. This is a high copy number plasmid, high copy number origin. And then the only other thing on this diagram I haven't explained is this, which is not relevant to us, and I even forget what that is, so don't worry about that either. If somebody's really interested, I can look it up, but I forget. Yes?
So is this how a usual passage would look like that we want to use in coli?
For expression, this is reasonably typical.
So all the MALI, and we would always do like a protein
Well, no, so I'm gonna show you some systems, so this is the one I like to use.
Okay.
And I like to use it because the MAL-E protects this other protein down here, so degradation of foreign protein is a real problem. It's less of a problem here because having the fusion protects your protein. You don't cleave it away from the MAL-E until it's out of the E. coli, right? We cleave it with the MAL-E in the test tube using this system here. This is after it's out of the test tube and we cleave with the factor 10, right? So I like it because it protects the protein. If you don't use a system like this, I often see a lot of protein degradation. But I'm going to show you some other systems, because again, no one system is going to work for every protein that you'd like to express. Okay? I like this one because it works really well. It's got this fantastic protein purification, but it won't work for everything. It's always worked for me, but I know it doesn't work for everything. This comes from NEB, from the BioWeb, yeah. They actually have an updated version of this vector that's even cooler that I'll show you, but we'll stick with this one for right now. The new one's a little more complicated, but even more cool. So I'll show you that later. Okay, so this is our fusion. We're making our gene fusion, which is going to result in our protein fusion, and then we're going to purify it this way. Okay. We have burned up a lot of time talking about that, so now we're going to go to the protocols, because I need to tell you how we're going to purify the protein. So